Hi, everybody. Thanks for the invitation, Jennifer, Catherine, Radhika, and a couple of disclaimers. I beg your pardon?
The computer hates it.
Oh, it doesn't like me. So, I'm not a climate scientist. I'm just a regular old microbiology guy. And so it's actually, I was thinking as Annie was speaking, it's actually, I'm making a plug for interdisciplinarity because what I'm going to talk about is my next foray, I guess, into the near future. But things I'm going to talk about are actually fungi, and I think you put this title on, right? I didn't give you the title. I think Jennifer did this, so I'm just talking now.
Michael's saying he's a fun guy.
Always. I apologize for the quality of this slide, but this is actually about fungi and emerging infectious disease, and this is from an article in Nature in 2012, and it's really right up Johnny's alley. But it shows quite nicely that if you look at the emergence, the green line represents fungal diseases in plants, and the red line are fungal diseases in animals, and how they've been increasing over the last decades. And then this figure shows the distribution of these various outbreaks over our globe. And this shows the red bar here is all the new fungal diseases that are emerging. And then this big bar over here shows the number of extinction events that have happened due to fungal infections across the globe. So it's really quite amazing. We talk a lot about viruses, we talk a lot about bacteria, but this is now a really, really emerging area. And in 2005, another plug for interdisciplinarity, Linda Allen, I don't know if Linda's here, she's a mathematician. She invited me to speak with her because she was writing an NSF proposal to the math biology program. And she said, hey, I want you to partner with me. And so she had me read this proposal idea that she had, and it was about a fungus. And I'm a microbiologist, and I hadn't heard about this fungus. And I was really embarrassed. But anyway, so I went in with Lawrence Smith. Some of you may know Lawrence Smith. He's now in Oklahoma. And the thing got funded, and so I said, oops, I better learn about fungi again. So I switched from my prokaryotic biology world into the eukaryotic world. I beg your pardon? I saw the light. Well, I did take 5 classes in graduate school in mycology, so, But so here are some of the examples of fungal diseases in the recent past that have kind of, in the last, say, decade and a half or so, that have really emerged. So we're looking on the far left panel, you're looking actually at a Magnoporta rhizae, which is basically a fungus that's on a rice leaf that's now emerging, and it's a big pathogen. And there are many, many fungal diseases on plants that are well recognized. It is those that are infecting animals that are less well understood now as emerging entities. And so on the far right is a fungal disease that's now wiping out all the frogs across the planet, and about 500 species have gone extinct already. So it's one of the greatest extinction crises that we have ever faced. That's a fungus that our lab works on, and some of my lab groups over there, I didn't know they were coming. And then on the bottom left, I don't know if you can see that very clearly, but it's the white-nosed bat syndrome, another fungal disease. One of the interesting features of the fungus that's killing the frogs and the fungus that's killing the bats is that they're both cold-loving fungi. Isn't that interesting? Okay. You just wait for the day they start switching over to warm-loving fungi, and then become our personal problem, So, and then the one on the right, you can't see that very clearly. I don't know if you can. Do you know what that is? The honeybees. Yes, and they've fingered there are multiple entities, but there's a fungus involved as well with that. And so here's where I actually talk about the atmospheric world, kinda. So on April 29 of last year, I was teaching my bacterial physiology class, and it was the third day when the wind was howling and we had brown sky and all of this, and I was really, really pissed off as I walked from my car to the building. And so I went upstairs, I think I called Amanda and said, bring me 12 Petri plates. So Amanda brings 12 Petri plates down, and I told my class to go stand outside for 2 minutes and just hold the plates up. And so that's what they did. And I put them down. About 48 hours later, this is what we saw. You can see there's a leaf that actually blew on there. So it's a real thing, right? And about 3 days later, that was the same plate, okay, at room temperature. So then what we did was we did some 16S ribosomal RNA sequencing, pyrosequencing, simply to identify who's there. You know, how many from the Hayhoe family, how many from the Vanels family, how many from the Jean family, and looked at that distribution. I have reams and reams of data, as you can only imagine, right? So we did a few plates and did that work with Scott Dowd. Scott's great. He just turned it around for me in no time at all. And as I began to look at the bacterial species and genera that were on there, I suddenly went, holy cow. That's a very Indian term, by the way. It was, you know, I was expecting to see bacterial species that were warm, loving, New Mexico, you know, Arizona, desert type microbes, one would think, right? Is that correct? So instead, and I'm not showing any of those data here, many of them, and the bulk of many of them were cold-loving bacteria, psychrophiles, some of which are found in the tundra, some of which are found in the Himalayan glaciers, all over the place. So I said, I don't know how to understand this, right? What's happening in the atmosphere? What was blowing on the earth that day around this environment? And so I know Jennifer. I got to know Jennifer, and so she's great. And so she said, yeah, let me look. So Jennifer gave me this. This is for all the atmospheric scientists. What did you call this graph?
High split.
A who?
High split.
A high split.
Graph.
Okay, so basically, if you look at the lower panel, we're looking at the movement of the air mass from the 27th of April, correct me, please, Jennifer, jump in, okay, through the 29th when we took our collection, okay, and I did the collection on the 29th of April. And so apparently, the air started off somewhere in California where it says start, okay, and they call it parcel, they call it a parcel of air. And I didn't realize it takes so long to blow across. That's the first thing that was kind of informative to me. So I'm learning atmospheric science now. That's my new interdisciplinary world. And then it kind of hit the Rockies and bounced up into
The
the
red line, we're thinking at the bottom and then the
So
top.
the red line just shows the general direction, right?
Yeah, so the top we're looking at the general horizontal direction, and then the graph at the bottom is showing our vertical direction. So the red line we're interested in because it's the one that finishes at the surface where we are and where the samples are taken. And so we can see that if we watch the red line at the bottom, it jumps up above 1,000 meters above ground level, which is actually really high. It was kind of went over Denver and the Rockies and stuff. So it got pushed up really high into cold, cold air, and then came down to us at the surface in Lubbock.
So what is curious to me was, you know, I never realized that there was all this mixing going on. And obviously there are higher level currents of air that are blowing, right, besides these. And so there must be some very interesting activity going on. And I was thinking, when I was looking at this, I was thinking of the flyways that the birds use. My daughter is, you know, working at Mono Lake, out in California, and also the Salton Sea. And in the last 3 years, she's noticed some huge changes in those water bodies because she studies those birds that migrate along those lakes and up to the Great Salt Lake. And so I was thinking about vectors and atmospheric transport of things besides the wind currents. So you can see as things change with climate, and if water bodies, large water bodies dry up and change, then those flyways are going to change, and then the atmospheric currents are going to impact things. So there's all of this stuff comes together in a very complex way. So anyway, I thought that was very, very interesting. So this is a picture of something taken on January 29th, so just last week when the wind was blowing. She called me. We've been taking samples all the way through, you know, last year through December before Christmas. We were on top of the roof. I had a picture I forgot to put of Jennifer holding a plate on the 12th floor of her building. My students love to come up there, by the way. And it's like a high place. Anyway, so we have two different Petri dishes, two different types of media. So the one on the left is a fungal stimulating medium called potato dextrose agar, and the one on the right is just a bacterial supportive medium, Diptyque soy agar, so we can get more fungi and more bacteria. So there's lots of fungi in the air. So on last Thursday, this is what you were breathing. by the way. Turns on my way home, I realized I take all the plates home, and and then I bring them to lab the next day. It's fine. It's not lab work, so. And I realized I had 2 plates left in the car that were unused that day. I do ground level things too. So I put my windshield down and I held the plate as I drove on the Marsha Sharp. So I'm going to do a separate analysis of that too. So when you have your your BMW with the top down and you're riding along, you may get a totally different perspective of the microbial world. I'll give you those data later. So, just a little bit about what we do in our lab is we are studying this fungus. We call it BD, Batrachocatrium dendrobatidis. This is the one that's been fingered as the major agent for causing this global amphibian decline in the last 20 years or so. And am I close to my 10 minutes?
Yeah.
I'm done?
Close, yeah.
Okay, close. Anyway, so we're studying this, and so my students are working on this. It's an amazing pathogen. A good pathogen is like a child. You don't want to kill your parents, because then all your resources are gone, right? So a pathogen that is opportunistic is like this organism, because it'll wipe out an entire lake or an entire water system of frogs in 2 weeks. Okay, so this is what happened in the Sierra Nevada, so what you're looking at. So we are seeing emergence of these opportunistic pathogens, okay? And that's a big issue, because they don't care, because they have an alternate lifestyle, and they don't care for the host they kill, because it's an easy prey. That makes sense? That's something to think about. Okay.
Sociopaths.
Like sociopaths, yes. So they kill, and so the things we're trying to study, and these fungi that kill this particular frog, we're trying to understand how it suppresses the immune system of the frog. It really does. People have shown that, and so we are trying to understand how it turns out that, so some of you when you all were young, you'd take a frog and put him on his back. What would happen? It would right itself again. That's called the righting reflex. Well, once they have an infection with this fungus, it loses that ability. So there's a neuromuscular issue as well. And so they're all very interesting things that are happening. So we're actually trying to understand a toxin that is getting produced, that's produced by this fungus, and try and figure that out as part of our studies. And we're also studying proteases and the ability to penetrate the mucin of the frog. But anyway, I'll leave you with that. And thank you. 10 minutes.
I have 2 questions, but I'll only ask one principally in the interest of time. If you go back to the dispersion plot that you did,
Yes.
the start to finish, I don't know how much you're suggesting, so perhaps this is the naivety of the question. When you held up these plates on the 29th,
Yes.
how do you know what, if the technical term, parcel of air you were actually
Sampling?
how do you know it was 2 days ago? How do you know it wasn't 1 day ago? You just know the parcel path.
Correct.
How do you know what was the starting parcel?
I'm going to leave that to the
We
atmospheric
can-
science.
Yeah, you can kind of set the model to say if you want 5 days out, so we could go 10 days out.
I realize that,
Yeah.
so are you, but.
Why did we decide to go 3 days out?
Yeah, and you're being highly suggestive that this particular sample that you collected really started 2 days ago and was not 1 day ago or somewhere.
Right.
I mean, how do I know
Sure.
that the parcel you collected really did that trip over up high and came down and didn't just come from?
From
the
Flomont.
Yeah, that's just relying on a model, but.
So in water, you can use like a dye or a chemical, right? So what do they use for like air? How
Yeah,
do-
but
how do
in
you
water you
track
get
it?
tremendous dispersion.
Sure.
I
Yeah.
presume you have this.
All these ADPO people are sitting.
Yeah, you're just using the vector property of some model that they
Yeah.
developed.
Yeah.
No, if you want to get more complex, you have to consider the boundary layer movement and surface particles versus particles that are at 500 meters above ground. So
Yeah, this is very simplistic and just kind of being like, okay, trying to figure out what's going on, and
Yeah.
there's so many other ways we can try and look at this, and different days out and different levels. We just chose those 3 levels of the atmosphere ambiguously.
Yeah, you're just doing a particle track.
We're exploring,
Right.
right?
Right.
Yeah.
I have one quick question.
Sure.
Can you show the slide of the 2 plates where you had more?
Oh, recently?
One,
This?
how did they differ? Where were they located that you had more fungi in one than the other one?
Well, these were at the same location. So, like I had my secretary and I, we just walked around my building, and then we did that. And then we did similar plates up in Jennifer's place.
But this one's bacteria.
So this is supporting mainly bacteria, and this is supporting mainly fungi. So you'll see some on both plates, but yeah. So when we do the analysis, we'll see some of the same, but we'll also see some big difference. And we haven't done any fungal analyses yet, so that'll be kind of curious.
In terms of the viability of the bacteria and fungi in the atmosphere and their atmospheric lifetime, what's known about that?
Well, in terms of the short time scales, that's not a big issue. Many of the bacteria that we found also are spore-forming bacteria. So endospores that bacteria form are the most biologically resistant structures on the planet beyond lignin. So that's not an issue for the most part. There are many bacteria that do not form spores. But I'm thinking over the 2 days that they're just flying around, there's something called viable but not culturable. So we don't necessarily capture everything that landed on the plate because some of them may be alive but they don't grow. So
At least for the fungal components, the darker spored ones have been shown to be intercontinental in their movement. The lighter spored, and most of those things that are on that plate are dark spored fungi, because that's what you tend to pick up. By the way, there's a number you should know. Every breath you take in, the average person breathes in 20 fungal spores.
yeah, and it's because our functional immune systems are active is why we can stand here and be able to breathe all those fungal spores. I mean, every time you take a step on the carpet, you're sending up a little puff of dust, right, full of skin cells and fungi. So
Don't start talking about our colon.
just let you know.
All right, thank you
All
very
right,
much.
thank you.